Good afternoon from North Wales in the UK. My name's Matt Rogan, and today I'm presenting a more detailed version of a poster I recently presented at the ISMRM 2021, titled Hypoxia Alters Posterior Cingulate Cortex Metabolism During a Memory Task, a Proton Functional Magnetic Resonance Spectroscopy Study. In this research, we will refer to environmental or polycapnic hypoxia. This is hypoxia resulting from a reduction in the fraction of inspired oxygen in the air we breathe into the lungs. Blood gas concentrations are free to vary with ventilation. There is no gas supplementation or clamping. This hypoxic stimulus is akin to hypoxia that ensues with high altitude pursuits. Upon exposure to hypoxia, the degrading arterial oxygen saturations trigger vasodilation and an increase in the global cerebral blood flow with the aim of maintaining the delivery of oxygen to the tissues. However, despite this increase in the global cerebral blood flow, on a regional level, this does not result in a uniform increase in perfusion across the brain. Posterior regions of the brain, particularly the posterior cingulate cortex and angular gyri, have been shown to have a reduced perfusion during hypoxia compared to normoxia. These regions form a major nodes in the default mode network of brain activity, suggesting that this observation may be specific to this network of spatially connected regions. The cause of reduced perfusion can be brought down to three main mechanisms. First, perfusion is reduced because the baseline or resting neurometabolic demands of those regions have been reduced in hypoxia. We would expect this given the coupling between neural regional metabolic demand and blood flow delivery. Second, the normal coupling between regional metabolic demand and delivery of regional cerebral blood flow is disrupted or reversed as a result of hypoxia. Or finally, another vascular phenomenon that occurs as a result of hypoxia and it happens to be that these regions are susceptible to this, and then this is then driving the reduction in regional perfusion that we see. In order to further investigate this, a study by Rossetti et al. Used two cognitive tasks that evoked opposing bold signal changes in these regions that at rest display a reduction in perfusion during hypoxia, namely the PCC, or posterior cingulate cortex, and the angular gyri. They compared the task-evoked bold response in both normoxia and hypoxia in these regions. So, if reduced perfusion in those posterior default mode network regions during rest is the result of a reduction in regional neural activity, upon functional activation of these same regions, because the baseline neural activity is lower, you would expect to see a larger change in bold signal in response to task and compared to the same change in normoxia because of the relationship between change in bold signal and the baseline bold signal. However, if instead the observed alterations are the result of a reversal in neurovascular coupling, the direction of the task-induced change in hypoxia will be reversed. As expected in normoxia, episodic memory recall resulted in a positive bold response within the regions of the default mode network, and visual motion attention task resulted in a negative bold response within those same regions. However, completion of these same tasks whilst being hypoxic reversed the direction of change in the bold response to both tasks, with episodic memory recall resulting in a negative bold response and visual memory search a positive bold response in hypoxia. The authors conclude that this observation is the result of a reversal of neurovascular coupling, with increasing neural activity demand triggering a reduction in blood flow within those regions during hypoxia. However, the bold signal is a vascular response that we use to infer changes in neural activity. In order to be sure that the reversal in the bold response we observe is the result of neural activity to task triggering a reduction in regional cerebral blood flow and not the result of altered vasculature alone as a result of hypoxia, we must take a direct measure of the task-induced neural activity. To do this, on the present study, we used a functional magnetic resonance spectroscopy to measure changes in glutamate metabolism during the same episodic memory recall task as Rossetti in both normoxia and hypoxia. Using this technique, we can predict that if hypoxia results in a reversal in neurovascular coupling, measured changes in glutamate concentration using fMRS during episodic memory recall will be the same in normoxia and hypoxia conditions. An alteration in this measured glutamate response to task during hypoxia would indicate that there is a change in neural activity or metabolism within the PCC. We scanned participants in both normoxic and hypoxic conditions on separate days. The procedures were matched completely and the participants were blinded to what condition they were in. We positioned our MRS acquisition voxel over the posterior portion of the cingulate gyrus. The average position is shown in the image below as a heat map over the MNI152 brain, and there's also a sample spectrum to assess our data quality beside that to the right. Our fMRS, functional MRS acquisition scheme was time locked to the individual trials within each block. This time locking of acquisitions allowed us to always acquire a rest shot 2 seconds prior to the start of a trial, and then a recall or response shot that was always acquired between the end of viewing the memory stimuli and the point at which the participants make the response. So in some cases, we would, if the individual took a longer period to respond, we would require an additional FID or shot here. But all of them would always have one which occurs straight after the presentation of the memory stimuli. We call this the recall period. Rest and response shots across each trial in all blocks were binned and averaged to create a single rest and response spectrum for each participant in each condition. We then compared the change in glutamate concentration between rest and response in both conditions. These were the same condition comparisons made in the GLM for the Rossetti fMRI study, what we showed earlier on. So what did we find? In normoxia, we measured an increase in glutamate concentration between rest and recall. However, this increase due to task was no longer present in hypoxia, with no significant change between the two. Additionally, behavioural performance on the episodic memory recall task was altered between conditions, with participant performance accuracy being reduced in hypoxia compared to normoxia. Further exploratory analyses of the other markers of metabolism did not show any task effects. However, glucose was seen to be significantly decreased in rest and response during hypoxia compared to normoxia. So, from our results, we conclude on the following. In normoxia, the measured increase in glutamate between rest and task demonstrates that functional MRS is an appropriate index of functional neural activity. This task-induced increase in glutamate we observe reflects an increase in oxidative metabolism and enhanced neural transmission. As a result, the loss of the task-induced increase in glutamate during hypoxia can be interpreted as either a reduction in neural activity to the task in that region, which our behavioural data showing reduced performance would support, or it could be a switch to non-oxidative metabolism within this region, the PCC. This is supported by the observed reduction in glucose during hypoxia, which we presume would reflect continued consumption within this region by non-oxidative means without sufficient delivery. We know that delivery of glucose via CBF will be reduced given that the PCC displayed reduced rest and perfusion during hypoxia. As part of this study, we actually acquired a whole brain rest and arterial spin labeling scan before we began the fMRS acquisition, so we could quantify regional perfusion, as been done in the past. And what we found was this also demonstrated as the earlier studies that I showed on the first slide, that the PCC after around 2 hours of hypoxia displayed this reduction in perfusion compared to that seen in normoxia. So that would support this conclusion of reduced delivery of glucose. Thank you very much for listening. The references used throughout are displayed below, and I'll accept any questions now.